Hello, what a pleasure to be here. I have been following the seminars and the work of many of the scientists in the Institute for a while, and I'm super happy to present our latest work on the temperature sensitivity of bat antibodies that we just recently published. I'm speaking to you from my home country, Bulgaria, and as a biodiversity hotspot, Bulgaria is home to some of Europe's largest bat colonies, making it an excellent place to do bat research and conservation. And bats are truly remarkable. They're the only mammals capable of powered flight, which indeed is the main driver of their rapid diversification into over 1,400 different species, which makes them the second largest group of mammals on the planet, and also the fastest flying invertebrates as well. This ability, however, comes with high metabolic demands, which they meet mainly through oxidative phosphorylation. However, this process comes with some costs, because during it they produce a lot of toxic byproducts which are causing DNA damage, and bats have encountered this by enhancing their DNA repair mechanism, which leads to their exceptional lifespan and even health span. Additionally, during flight, their body temperature rises up to 42 degrees, which mimic a fever response and create a selective force for the pathogen coexistence. On the contrary, during their resting phase, bat's body temperature can drop to 20 degrees Celsius. In hibernation, it can fall even below 10 degrees for days, weeks, and even months, which significantly decrease both their innate and adaptive immune functions. And it has been suggested that actually activity during the winter is a key reason. It has been suggested that activity during the winter is mainly for immune activation and not so much for feeding, for example, because during hibernation their temperature is low, so they experience prolonged fasting, and their metabolism is very slow, and this is also likely to slow viral replication and even host cell division. And those seasonal and daily body temperature changes are yet another interesting aspect in understanding their evolutionary immune responses. They have other unique traits like roosting in large groups. There are many different bat species, and they have high pathogen tolerance, which makes them an important reservoir for zoonotic diseases as well. And to understand their evolutionary advantages, we must develop experimental setup that incorporates multiple aspects of their biology. And additionally, while temperature is a crucial factor for the immune system functioning, this area is still largely unexplored, and it's not only for bats, even for us humans as well. Unlike the innate immune system, the adaptive immune system can recognize specific pathogen and create a memory response in order to fight future infection in more effective ways. B cells are producing antibodies that have two main parts: the FAP region, here the fork, which is the one that binds to the specific antigens, and the FC region, so this part here, which drives the immune response. And although bat immunity has been a hot topic recently, most research is focusing on their serological, like just doing serological screening and genetic extrapolation. So we know very little about their humoral responses, and we don't really understand the specific of how bat antibodies work, even though they play key role in the defense against pathogen and maintaining immune balance overall. And thus we decided to create an experiment and track how fluctuation in body temperature affect the adaptive immune compartment in bats. And we also look the role of this temperature fluctuation in terms of inflammation responses and keeping the homeostasis. To tackle this, we collected blood samples non-invasively from several bat species across different locations and colonies in Bulgaria. And all the captured bats were rewarded with mealworms after the end of the treatment, so they were happily released. And we focus primarily on two species that has a relatively larger body size. So this is the greater mouse ear bat and the common nocturnal bats. So their body mass is around 30 grams during the active season, which allow us to collect sufficient amount of samples. Also, the two species are quite abundant. They're easy to capture, and we have done multiple prior research in the colonies. We were collecting samples, so we knew a lot of other things about the animals. So once we collect the serum, we purify the IgG, and then we're testing their reactivity towards huge panels of antigens at the three significant temperature that they experience during their daily cycle. So 4 degrees, which is kind of more the deep torpor state, 22 degrees, which is more the shallow torpor state that they enter during the day when they're resting, and 42 degrees, which is kind of the extreme active state when they're flying. And we conducted multiple ELISA experiments and also self-sorting experience in order to see how these antibodies are behaving, what are they doing, how they're doing, and why they're doing. Then as a control, we have serum from healthy humans, then cattle, mouse, goat, rabbit, and another 4 bird species in order to see if any temperature sensitivity is also present in other species than bats. So first we wanted to explore if anyway temperatures has any effect on the diversity of bat antibody antigen specificity. So at 4 degrees and 22 degrees, bat antibodies show negligible reactivity, as you can see here on the heat map. And this reactivity was significantly broadened at 42 degrees. They start to react with way more of the antigens, and but some of them they were binding preferentially. So you can see that some viruses, like some proteins of the tick-borne encephalitis virus and rabies, were really like extremely increasing their binding affinity when the temperature was higher. And this was not true for none of the human samples we tested, or none of the other samples we tested from other mammals and bird species. Of course, with birds, we were working with IgY, which is a bit different, but we wanted to see, because they're also flying animals, if this is something to do with flight in general, or it's something else. We then did the same experiment, but not with purified IgG, but with the full serum, which is kind of a more complex system where the IgG might behave differently because there are other things around, and it was exactly the same. So we did so many ELISAs and everything was always the same. Yes, temperature affect their diversity. Next, we use surface plasmon resonance to measure the effect of temperature on the antigen binding properties of bat antibody, and this method allows for a precise measurement of the interaction strength, the binding kinetics, the specificity, and also the optimal temperature at which the affinity is the strongest. And we focus on the two antigen I pointed out from the previous experiment, the NS protein of the tick-borne encephalitis and the rabies virus glycoprotein, because they show the strongest effect. So we first wanted to test with the best case scenario, and antigen were immobilized on a sensory chip. Then antibodies were injected at various concentration, and further we could analyze how temperature is affecting the binding. And you can see that at 4 up to 16 degrees, the binding is actually minimal, almost no binding happen at all. But when we increase the temperature at 40 degrees, which is kind of the maximum, the limit of the machine we're using, the interaction was really strong, and it was concentration dependent. Then to gain a deeper insight into how temperature affects bad antibody repertoire in general, we use a protein array, which is displaying over 70,000 human proteins. And we tested again the same serum of the same group of bats at 22 degrees and 42 degrees in order to see if there are plenty of opportunities and plenty of different things, would this effect be the same? And as you can see from the graph, at 42 degrees, the repertoire of recognized proteins significantly broadened, and binding was strongly with more than 2,400 different pathogens, so about proteins, so about 40% of all the present proteins. However, at 22 degrees, the same antibodies were recognizing only 19 protein. And in contrast, the human IgG was not showing such a significant effect. Of course, there is a little bit of an effect, but not as strong, definitely not comparable with the one we were observing with bat IgG. And our data suggests that bat antibodies significantly increase their antigen binding diversity at high temperature, and which is typical for the active metabolic state. Then we ask another question: are there more polyreactive IgG in bat serum? Because polyreactive antibodies are known to some of them to be more temperature sensitive. Even though there is only one publication that did any experiments on that, and it was a very old study, and we use a very commonly used method with dinitrophenol conjugate in order to sort the polyreactive with monoreactive antibody in the serum. And as you can see on the graph, bat serum is having way more polyreactive antibodies than human serum with the green line. And in humans, it's about 20% of all the antibodies in the serum are actually polyreactive. So probably in bats, this percentage is way higher. To understand the molecular mechanism of this antibody diversification, we study their molecular properties, and we use a thermal shift assay in order to see the melting, the lower and the higher melting point of the antibodies. You can see that but antibodies are showing lower melting point compared to human antibodies, which means that they're less thermally stable, and this in a way can explain their temperature sensitivity. We also take test the hydrophobicity, but it seems that they're similar in this parameter. And then we wanted to investigate actually the physiological role of temperature sensitivity, and we tested the capacity to opsonize a human endothelium cell, and the increased metabolic rate during flight is also releasing a lot of ROS, which cause cell death and pro-inflammatory activity. And but antibodies at 42 degrees might, so during flight, they might help clear those damaged cells. So we model oxidative stress by exposing human endothelial cell to hydrogen peroxide and incubated again but antibodies to different temperature. And then we extended this experiment, including also bat cell lines as well, in order to see if what we're seeing in the human cell line is just because the cells are too different, and that's why they're so reactive. Because you can see here that their antibodies are reactive even to live, but their reactivity towards dead cells at 42 degrees is so much bigger. And all of this suggests that bat antibodies help in the cleaning of dead cells process, and this probably happened during the active state of the metabolism, and is reducing inflammation in general. So our results directly link their metabolic states and their immune function. lower antibody reactivity at low temperature may conserve energy during torpor, while increased reactivity at high temperature might provide an immediate protection when it's needed the most. And this rapid response covers a broad antigenic space, which is very good because you don't need so many specific antibodies and you can act with whatever is there. But even though we saw that some of the temperature-dependent specificity is actually also probably shaped by co-evolution with certain viruses that we know that are present in the study population. And in addition, but IgG preferentially binds to dead cells at high temperature, aiding in the clearance of damaged cells and reducing inflammation. So understanding how this process has evolved can shed light to the evolution of their unique immune system, and also can lead to the development of human temperature sensitive therapeutics. And we published already this study, but we want to increase our sample size, use many different bat species that, for example, are not they don't hibernate, or also use other mammals that their natural temperature is also higher. So there's some mammals that their temperature is 42 degrees without flying. So it will be really interesting to get a deeper insight into this evolutionary process. So if you have any serum or you would like to collaborate, we'll be really happy to get in contact with you. And with this, I would like to thank you for your attention, and I'm happy to answer your questions.